Good afternoon, everyone. I'm Madhulika. I'm a postdoc in Yadavalli Lab at Rutgers University.
My name is Arpita. I'm a graduate student in the Yadavalli Lab, also at Rutgers University.
And today we'll be talking about filamentation in E. coli. So our lab works on understanding bacterial regulatory networks and stress response mechanism, focusing on epitranscriptomic factors and small protein. Today we'll be talking about role of one epitranscriptomic factor in bacterial stress adaptation. Bacteria have to adapt to diverse stress environment, and signaling system help detect these changes. One of the signal transduction in bacteria mainly occurs through two-component system, and one of the well-studied two-component system is the PhoQ system. It consists of a sensor histidine kinase PhoQ present in the inner membrane, which senses the external environment, and the second component is the protein PhoP, which gets phosphorylated and drives the expression of downstream genes. PhoQ system is important for magnesium homeostasis, acid resistance, and resistance against cationic antimicrobial peptides via lpase modification. Bacterial filamentation is now also associated with stress adaptation, as opposed to the traditional notion that considered it as a hallmark of cell death. One example is uropathogenic E. coli that causes urinary tract infection. Filamentation helps UPEC colonization, as filamentous cells evade phagocytosis by immune cells better compared to the shorter cells. Our lab has reported that in presence of certain stressors such as antimicrobial peptides, there is strong activation of the PhoQ system, which causes E. coli cells to filament. And this phenotype is reversible once the stressor is removed. These filamenting cells are viable and they show no obvious growth defect. We believe that it is a protective mechanism against stressors that strongly activate PhoQ system. My colleague Arpita will elaborate on it.
So to follow up on what Madhulika has already told us so far, when there's a strong activation of PhoQ, for example, through the addition of antimicrobial peptides, that leads to a cascade of responses that occurs by increasing the phosphorylation of PhoP, and this releases into an increase of let me see. Okay, an increase of QueE, and this causes a substation inhibition via divisome binding that leads to something called filamentation here that you can see. And now we know that QueE localizes to the Z ring, which marks the site of cell division and is noted to be important for filamentation. Therefore, it is highly likely that QueE has some direct interaction with a component of the divisome that leads to this filamentation. But before I get into that, I first want to talk briefly about QueE. Now, QueE is an enzyme that is important for the biosynthesis of cuisine tRNA modification. And this cuisine tRNA modification is important in translational accuracy and efficiency. What we know is that under strong upregulation of stress responses, such as the introduction of antimicrobial peptides, we note that the QueE release is involved with high cuisine tRNA modification, but it also plays a role in inhibiting cell division. And here in the model that we see to your right, it shows residues that affect both Q biosynthesis only, and in red we can see residues that affect cell division only. Now, what we know is that there's a dual function of QueE in the cell, but again, these proteins and these residues were found by alanine screening mutagenesis, and what we note is that we found this through microscopy. And further noting more, I would like to talk a little bit more about the E. coli cell division pathway. This is a highly organized process that can be involved in 3 different stages: the early stage, the mid stage, and the late stage of the proteins. We know that FtsK is the first protein that is recruited sequentially to the site of cell division, which is known as the Z ring, and then subsequent proteins follow and are recruited to this site of cell division. We also know that there is furthermore peptidoglycan synthesis and hydrolysis. And from this, we'd first like to see that since we know that there are other proteins that are also involved in cell division, we first want to see that QueE, which is this protein that we assume is involved with filamentation, we want to see whether this is involved and recruited to that site of cell division. So first, using FtsZ as a marker, because we already know that this is found at that site of cell division, QueE and FtsZ, when merged through these microscopy images, show that they're both found at that septum or that location where cell division occurs. So we know that this is recruited to that site of cell division. This gives us a great basis to ask our next questions. So first, we want to ask, what is the mechanism of QueE-mediated filamentation? And in order to answer this, we want to first find our interacting partners of QueE that are found at the divisome. Of course, this is great, and I would like to show you all images in the next upcoming slides from our microscopy experiments that co-localize the proteins of the cell division with FtsZ in order to see whether they are found sequentially and if they are recruited fine when they are stressed under upregulation of QueE. When looking at these early proteins, when there is QueE upregulation occurring, what we note is that the proteins are recruited fine to the site of cell division, as FtsA and ZipA are both found at the septum, as they should be, co-localizing with FtsZ. However, when we look at the late division proteins such as FtsQ, FtsW, and FtsI, what we note is that all three of these proteins do not co-localize at the septum, which means that something is occurring prior to these steps. So now we have a clue as to where the problem might be occurring. Now, we know that the early cell division proteins are recruited just fine to the site of cell division, and the late stage proteins are not recruited fine. Therefore, we can assume that there is something happening prior to these steps, or the mid-stage proteins such as FtsK. However, FtsK is one of those proteins that's an essential cell division protein, but it's very complex to work with. Structurally, according to the literature, the N-terminal domain of FtsK is involved with recruiting the late-stage division proteins. And we know that the middle of FtsK is involved with linking both the N-terminal and C-terminal ends, but its exact role is unknown at the moment. And we know that the C-terminal domain is involved with forming a little spherical structure that helps bring in the DNA right before cell division occurs. Now, with all of this given, FtsK is a very, very difficult protein to work with, whether it's creating a construct or to even observe this under fluorescence microscopy. But one lab was actually able to create a construct. And when we took a look at it under the microscope, it gave us some pretty interesting results. Compared to the early division proteins, so these are the ones I showed earlier that recruit just fine at the septum at regular intervals, what we found is that FtsK preliminarily appears to only localize at irregular intervals. So that means that something is occurring that's inhibiting its location to the cell division site. So we want to further test whether this is occurring by co-localizing with FtsZ, and also to use bacterial 2 hybrid assays to further validate our results and hopefully find whether this is the protein that is being targeted directly or indirectly.
So we know that during QueE-mediated filamentation, the early divisome proteins are recruited fine, but the late divisome proteins are not. And if this is the case, then in presence of antibiotics that target the late divisome protein, the filamenting cells and the non-filamenting cells should not be affected to the same extent. To test this hypothesis, we use 3 different antibiotics. Ciprofloxacin targets DNA replication, which is a step way ahead of cell divisome assembly. Cephalexin targets the late divisome protein FtsI, while phosphomycin targets murA, which is involved in the first step of peptidoglycan biosynthesis. So starting with phosphomycin treatment, the phase contrast micrograph here shows the morphological state of the cells, that is whether they are filamenting or not filamenting. And we could see that filamenting cells are more tolerant to phosphomycin treatment compared to the non-filamenting cells. And similar results were also obtained for cephalexin treatment. In presence of ciprofloxacin, filamenting and non-filamenting cells had similar growth. So this corroborates our data from localization studies that late divisome protein recruitment is affected during QueE-mediated filamentation. So coming back to this pathway, we know that our results so far suggest that QueE possibly affects the localization of FtsK, which is in the middle of the pathway. What we also know about FtsK from literature is that it interacts with an auxiliary protein, RlpA, which is an outer membrane lipoprotein important for maintaining the rod shape of the cell and cell envelope integrity during cell division. And the interaction between FtsK and RlpA is physiologically important because deletion of RlpA suppresses the growth phenotype and filamentation phenotype associated with an FtsK mutant. So we asked the question that whether RlpA has a role in PhoQ dependent filamentation. So as can be seen from the phase contrast micrograph here, deletion of RlpA suppresses QE mediated filamentation at later stages of growth. We also quantified this data, and the average cell length reduced from 25 micron for filamenting cells to 7 micron for filamenting cells with RlpA deletion. So we have this model for now where in presence of certain stressors, when the PhoQ system is strongly activated, there is increased expression of QE in the cell. This excess QE interferes with the localization of the late divisome proteins, but not the early divisome protein. QE possibly blocks the interaction between RlpA and FtsK. This impairs the localization of the late divisome proteins at the Z ring, causing the bacterial cells to filament. So we are presently localizing the FtsK and RlpA protein in filamenting cells when QE is upregulated. We are also looking at the interaction between QE and FtsK, and we'll be investigating the amino acid residues in QE that are important for its interaction with divisome associated proteins. With that, I would like to thank all the members of Yadavalli Lab and TSS for providing the opportunity to present this work. I'll be presenting this work in more detail in poster number 6. Arpita will be presenting right next to me on on effect of cationic antimicrobial peptides on PhoQ system. And there is another poster from our lab on small proteins by Nitish. It's poster number 45. If you have time, please check it out. Thanks. And any questions?
Any questions?
This is just a general curiosity question. You said that the filamentous form happens when the cells are stressed.
Yes.
If you remove that stress, do the filamentous cells then separate and become single-celled, or is it just that the new cells will become single-celled?
So these filamenting cells, they have multiple nucleoids and they have like continuous cytoplasm, and so there are equally placed Z rings. So as soon as the stressor is removed, that Z ring will, all the divisome associated protein will come to the Z ring and the cell septation will occur. So it's just that the cell has to septate.
That's super cool. Thank you.
I can also elaborate on your question a little bit. I actually performed an experiment where I did stress the cells by giving them a deficiency of magnesium in the medium. And then after 24 hours, I gave them another medium that contained all the nutrients that they needed, including magnesium, to the physiological conditions that they needed for growth. And I saw that all the cells became the short, normal cells that you would expect E. coli to be after that time period. So they will go back to that once the stressor is removed.
Nice talk. So if I understand your model correctly, so QE, you think QE is interacting with FtsK. So in some ways that interaction is it's interfering. So can you, and I don't know if this is possible, but can you, if that's true if you increase QE and that blocks normal cell division, can you overexpress? So in other words, is there a competition here, and then can you overexpress the other protein to basically outcompete the competitor and suppress the phenotype that way?
For FtsK, we cannot overexpress because it will be toxic for the cell. People have shown that even more than one copy can cause toxicity. So this protein, we haven't tried that with RlpA here. So yeah, we can try that.
Let's move on to the next talk. Thank you.
You're good.